So good morning everyone. Can you hear me, students? So this topic, regarding this topic, viral hemorrhagic fever, it's a very huge topic. And so regarding this viral hemorrhagic fever, it usually it causes severe multi-system syndrome because it involves a lot of systems. It causes damage to the overall vascular system. That is why hemorrhage is occurring, and the symptoms are often accompanied by hemorrhage. Okay, so these are the characteristics of this viral hemorrhagic fever. So viral hemorrhagic fever, there are distinct families. Here it's written the four distinct families. Most of the viruses which causes this viral hemorrhagic fever, they are Filoviridae, Rhinaviridae, Bunyaviridae and Flaviviridae, and to a lesser extent they are caused by Togaviridae. So these main ones, the main four ones, are the ones which actually causes this viral hemorrhagic fever. Fever means the viruses which belong under this group. So let us see. Okay, so regarding the filoviruses, you've heard of Ebola. Okay, Ebola hemorrhagic fever and Marburg viruses. These are the most dangerous among these viral hemorrhagic fever viruses. Okay, the others include members from the arena viruses like Lassa fever, Junin virus, Machupo. Then Banya viruses, that is Rift Valley fever, Crimean Congo hemorrhagic fever, and Hunter virus comes under Banya viruses. And among the flaviviruses, okay, which are included in this family are the yellow fever, dengue, Kiasanur forest disease, and chikungunya virus is included under Togaviridae. So regarding these viruses which cause this hemorrhagic fever, okay, all are RNA viruses. They are all enveloped, means they have a lipid coat. They are geographically restricted by host. Then the rodents and arthropods, in most of these viruses you see that the rodents are involved, the arthropods are involved. So they constitute the main reservoirs in these hemorrhagic fever viruses, and the humans are not the natural reservoir, and the ticks and mosquitoes are the vectors for some of the viral hemorrhagic fever. So this is in general regarding the hemorrhagic fever group of the viruses. Now they are highly infectious after aerosolization. So the infectious dose in some of these can be as low as 1 to 10 organisms. That's why they are very, very dangerous. And risk of person to person transmission, it depends on the virus. So the risk means person to person transmission, it's not necessarily very high risk for all the viruses, but there are some viruses which constitute a high risk group as compared to the other viruses. Okay, for example, look at the risk of human to human transmission. It is seen that there's none in case of yellow fever, Rift Valley fever, Hunter viruses. It's comparatively low in case of Lassa viruses and South American arena viruses, and very, very high in case of the major hemorrhagic viruses, that is Ebola, Marburg and Crimean Congo hemorrhagic fever. Okay, so regarding the pathogenesis of viral hemorrhagic fever, usually the cells which are involved are the monocytes, the macrophages, the dendritic cells, hepatocytes, adrenal cortical cells and endothelial cells. These are the main target cells. Okay, so once they enter into the human body, the initial replication occurs in the monocytes, the macrophages and dendritic cells, and via these cells transport these viruses to the different parts of the body. Okay, so they disseminate to the lymph nodes, the main organs like the spleen, liver, as well as the other organs. So finally, these infected macrophages and dendritic cells, they get activated. Okay, so the primary mechanism in most of these viral hemorrhagic fever group of viruses are that they release a lot of cytokines. Okay, so these infected macrophages and dendritic cells, they get activated. They release pro-inflammatory cytokines such as tumor necrosis factor alpha, interleukin 6, interleukin 8, etc. So these cytokines are involved with an increase in the endothelial permeability and cause vascular damage. So as a result of these cytokines, there will be an increase in the vascular permeability, and there's a vascular damage, and as a result of this, the hemorrhage usually occurs. Okay, so they, in addition to all these cytokines, there are additional there's viral glycoproteins such as glycoprotein 1, 2, okay, and BP40, which are associated with the virus, and these are associated with endothelial permeability by triggering expression of the adhesion molecule. So they facilitate, these viral glycoproteins actually facilitate or enhance the adhesion of these viruses to the host cells. Okay, so what happens as a result of all this? There'll definitely be endothelial damage. There'll be apoptosis of the lymphocytes. Okay, lymphocytes supposed to be protective, but instead they're being destroyed. There'll be hepatocellular necrosis because there's a decrease. As a result of this hepatocellular necrosis, there'll be a decrease in the clotting factor. It'll cause dysfunction of the coagulation system. Okay, and the adrenocortical necrosis also occurs. As a result of this, there will be hypotension, there will be decreased steroid synthesis, which can lead to hypovolemia and shock. And finally, the end result in most of the hemorrhagic viruses, the severe ones like Ebola, Marburg, will be a multi-organ failure and shock. So this is regarding the pathogenesis of the viral hemorrhagic fever group of the viruses. Okay. Now you can see the mechanism is either direct vascular involvement plus mediator induced vascular instability, primarily tumor necrosis factor alpha. Okay, it can trigger the coagulation cascade. So it not only acts directly, this virus, okay, it has a direct effect, but it also enhances or it also triggers the release of cytokines, which actually contributes to the pathogenesis of these particular group of the viruses. So why these viruses also have a huge potential for bioterrorism. Okay, so why these agents? Because these agents, they have a high potential under this bioterrorism group of viruses. These Ebola, Marburg, okay, and Lassa virus, they actually belong to the category A of these, means they are category A agents, okay, whereas there are some like Hunter viruses, they belong to category C. Okay, so why these agents? They are these agents have a great potential for bioterrorism because they have a high potential for adverse public health impact. They have a serious potential for large scale dissemination. Okay, so vaccines are unavailable in most of these viruses. Vaccines are not available. They are very easy to produce and easy to disseminate because they have a high risk of aerosolization. Okay, so they are easy to disseminate. So coming to the Filoviridae group, okay, these are the main groups which actually causes a very high mortality. Among this group is, within this group is Ebola virus and the Marburg hemorrhagic fever virus. The Marburg was first identified in 1967 in a European laboratory, workers in Marburg, okay, which is in Germany and Yugoslavia, but it also affects Uganda, Western Kenya and Zimbabwe. Means this Marburg viruses had caused epidemics in these countries. Okay, so Ebola and Marburg, the host or reservoir is unknown, but the human transmission can occur through direct contact with the blood, secretions or organs of infected persons, contact with contaminated needles, or contact with the reservoir or the secondary host. Even though the host or reservoir is unknown, but there may be reservoir which they don't know. Okay, so they assume that contact with such reservoirs may cause the Ebola and Marburg hemorrhagic fever. Okay, so these were the countries which were affected by which had outbreaks of Ebola. Okay, it started in the year 1976 in Democratic Republic of Congo, which was formerly known as Zaire. Okay, and since then it has caused epidemics in most of the African countries. Okay, so regarding the virus, Ebola virus, incubation period is 2 to 21 days. The clinical features are fever, okay, severe prostration, okay, exhaustion, diffuse maculopapular rash by day 5, and a distinctive bleeding and disseminated intravascular coagulation, which is very common. And the mortality can go up to 90%. Okay, whereas Marburg, the mortality can go into up to 70%. Okay, so even Marburg has the same type of clinical features in comparison to Ebola. So they are very, very similar. Okay, so this is maculopapular rash, which is seen in Marburg disease, seen on the face, the arms, okay, the head area. Okay, so coming to Ebola virus. Okay, Ebola virus is very, very important because of the recent outbreak. So it was first discovered in 1976 by Peter Piot, who was a Belgian microbiologist. He identified this Ebola virus. So he found that he saw a very peculiar looking virus which looked like a gigantic worm, okay, and very, very similar to the Marburg virus. Okay, later it was identified that it is different from the Marburg virus, and it was given the name Ebola virus. So it was named after the river Ebola in the Democratic Republic of Congo, where it was first found. It is often fatal in humans and non-human primates. Okay, it was fatal in non-human primates like the gorillas, chimpanzees, monkeys, and the first outbreak occurred in Zaire, or the Democratic Republic of Congo, 1976, where there were a huge number of human cases, which was a bit peculiar at that time. Okay, so 88% of those people who were actually infected with disease, they died. So there are 6 subtypes of Ebola viruses: Ebola Zaire, Ebola Sudan, Ebola Thai Forest or Ivory Coast, Ebola Reston, Ebola Bundibugyo, and Bombali virus. So with the exception of this Ebola Reston, all these strains have been known to produce infection in humans. Okay, but this Bombali viruses, they are not yet sure, because it was identified in bats. Whether it's causing disease in humans or animals, it's not very sure. Okay, so it was believed that the first human infection, how did infection with Ebola occurred? It was believed that it was through contact with an infected animal. Okay, because they found that in an outbreak of Ebola Zaire, which occurred in Gabon in 1996, it occurred after people had eaten an infected monkey. Okay, monkeys are known to be the host for these viruses, the Ebola virus. And one scientist who had conducted an autopsy on a wild chimpanzee in Thai forest in Ivory Coast was diagnosed with Ebola, Ivory Coast, in 1994. So they believe that the first human infection occur as a result of contact with an infected animal. So it also has a psychological toll, because it not only causes a lot of problems for the villagers, but they were looking for scapegoats, okay, to blame it on. So for example, there was a lot of unrest, a lot of uh, a lot of accusations against people. Okay, and for example, us means in 2003 they have stoned to death 4 teachers, and they believe that these teachers who actually were accused of casting an evil spell to cause this outbreak of the Ebola disease. Okay, because it killed many, many people. So the fruit bat are the reservoir for both Ebola and Marburg viruses. Okay, later they found out. Okay, these are the reservoir for both Ebola and Marburg viruses, and the hammer-headed fruit bat is a most important fruit bat. Okay, initially they didn't know the reservoir. Okay, they were not sure, but later on they found that the fruit bats, they are the reservoir for both the Ebola and the Marburg viruses. Okay, so spillover from bats is believed to cause the outbreaks, and the mammalian species, including the non-human primates like the gorillas, chimpanzees and all, they constitute the dead-end host. So modes of transmission: contact with the bats or the reservoir, as seen in hunters, miners, ecologists, tourists. It can also be caused as a result of contact with the secondary host or consumption of bushmeat. Okay, so through contact with infected animals, and contact with the patient, okay, with human in patient, with the humans. Okay, as seen means contact with the patients, as seen in deaths which occurred as a result of contact with the patients. The deaths have occurred in family members or as a result of nosocomial infections and burial practices which are improper. Okay, so you can see that the pathogenesis, human to human, it can occur in healthcare workers, in close contact of patient or contact with the dead body and infected needle, and reservoirs the fruit bats. Okay, and zoonotic, it's as a result of close contact with infected animal, as a result of butchering, eating of the animals, handling of the contaminated organs or the cells of the animals. So it is not transmissible prior to onset of symptoms. The virus load is maximum 9 to 10 days after onset of symptoms. And if the patient survives to day 14, there's increased chance of survival. Okay, so patient usually do not survive within 2 weeks. Okay, so but if they do survive, means there's an increased chance of survival, and the convalescence or the resolution of the viremia can occur henceforth. So we had an outbreak as recent as 2014. Okay, regarding this outbreak, what is means the information regarding this outbreak, the epicenter was in Guinea, that is West Africa, and the index case was he was a resident of Miliando village of Gekedo and died on 6th of December 2013. Okay, it was finally confirmed by WHO in March 2014, and the outbreak spread to Liberia, Sierra Leone and Nigeria, and subsequently outside of West Africa as well. So why was it alarming? Why was this outbreak alarming? Because it's the largest and most widespread outbreak since it was discovered in 1976. More cases and deaths in this outbreak than all the others combined. Okay, so multiple countries were affected, starting in Guinea, spreading across land borders to the other countries. And what is even more alarming is that these countries, they have a very weak health system. They lacked human and infrastructural resources, and they have only emerged, that recently emerged from long periods of conflict and instability. Okay, so this has led to an increase in the number of deaths as a result of this outbreak. So what are the treatments being evaluated for this Ebola? Okay, they've evaluated certain treatments, and they found that there's some good results have been found with human convalescent plasma, as well as a cocktail of monoclonal antibodies against Ebola, which is known as ZMapp. And there are certain nucleotide analogues which have been used, like favipiravir, BCX4430, brincidofovir, and small inhibitory RNA was also the treatments which were being evaluated for this Ebola virus. Okay, this candidate vaccine, the RVSB Zibov vaccine, it was licensed to Canada. Okay, and was a vaccine. It was a candidate vaccine then, but it has been used in certain countries, like Asian countries like the Philippines, Indonesia, and all, but not used in India. Okay, I mean in the I'm sorry, it was used in Ebola outbreak in Democratic Republic of Congo, but not used in other countries. Okay, so the candidate vaccine, it uses a vesicular stomatitis virus which carries a non-infectious Ebola virus gene. Okay, so the vaccine was being used in the ongoing Ebola outbreak in Democratic Republic of Congo, but not in the other countries. So coming to the next virus, which is very, very important, that is the dengue virus. It is a single-stranded RNA virus. It belongs to the family Flaviviridae, and there are 5 serotypes of dengue. Okay, dengue 1, 2, 3, 4, and 5. Okay, so knowing these serotypes of dengue is very, very important because it contributes a lot to the pathogenesis of dengue hemorrhagic fever. Okay, so it was dengue 1 was first found in Hawaii in 1944. Okay, dengue 2 in New Guinea 1944, 3 and 4 in Philippines in 1956, and dengue 5 was found as recently as 2013 from Bangkok. So viral replication, it occurs in the cytoplasm. Okay, so history, dengue fever was first referred as water poison, means it's a old fever. It was first referred to as water poison, and it was associated with flying insects in a Chinese medical encyclopedia in 992. Okay, and the word dengue, it comes from the Swahili word for bone breaking fever, or the word for the walk of a dandy in Spanish. And it was Benjamin Rush who coined the term breakbone fever because of the symptoms of myalgia and arthralgia in 1779. Okay, so the 4 dengue viruses, okay, originated in monkeys, jumped to humans in Africa and Southeast Asia between 100 to 800 years ago. It was geographically restricted, but because of the World War, it has spread to other countries. And the first clinically recognized dengue epidemics occurred almost simultaneously in Asia, Africa, and North America in the 1780s, but it was first documented only in the 1950s during epidemics in the Philippines and Thailand. So dengue is actually an emerging disease. Okay. So about 2.5 billion or 40% of the world's population live in areas where there's a risk of dengue transmission. WHO has stated that 50 to 100 million infections occur per year, okay, including 5 lakh dengue hemorrhagic fever cases and 22,000 deaths, mostly among children, and the average case fatality in dengue is 5%. So these are the countries which are at risk of dengue transmission, which are highlighted in yellow. So regarding the scenario of dengue in India, all the 4 types exist. Okay, type 1, serotype 1, 2, 3 and 4. And the first recorded epidemic was in Madras in 1780, okay, now known as Chennai. And there was a double peak hemorrhagic fever epidemic. The first time was seen in Kolkata between July 1963 and March 1964. And in New Delhi, outbreaks of dengue fever have been reported. So Delhi had seen a lot of outbreaks of dengue fever, and the latest was in 2015, which recorded the worst outbreak since 2006 with over 15,000 cases. So there was a lot of deaths as a result of dengue fever in Delhi in 2015. Okay, and here they found that the serotypes which are primarily involved in this outbreak was dengue 2 and dengue 4. Okay, regarding the dengue transmission is bite of infected Aedes species mosquito, but it can also occur through transfusion of infected blood or as a result of transplantation of infected organs after occupational exposure, as for example needle stick injury, and even cases of vertical transmission have been described, means from the mother to the fetus. Vector is Aedes aegypti, okay, which is actually a nervous feeder because it takes only a small amount of blood from a patient and it keeps biting multiple, means it takes multiple bites before it actually becomes full. Okay, it becomes satisfied. It takes multiple bites from different individuals. Okay, so the others are the different Aedes polliniensis, albopictus. Okay, these are day biters and they breed in clean water collections. So the dengue transmission, it bites the mosquito, the infected mosquito will bite the dengue. Okay, the uninfected mosquito will bite the dengue infected person. Okay, this mosquito, it takes 10 to 12 days for dengue virus to incubate inside the mosquito. Okay, and finally this infected mosquito will bite another person. This person will have the symptoms of dengue 4 to 13 days later. So we can see here that the mosquito, it actually transmits the dengue virus to human dendritic cells. The virus target areas, okay, with high WBC counts, for example, livers, spleen, lymph nodes, bone marrow, and the glands. The virus is then released from these tissues. They spread through the blood to infect the white blood cells and other lymphatic tissues. And then again they are released from the tissues and circulate in the blood. So in humans, this is a very, very important slide. Okay, you have to remember this slide. In humans, recovery from infection by one dengue virus, it provides lifelong immunity against that particular virus serotype. And evidence points to the fact that sequential infection with a different serotype increases the risk of developing severe dengue more when serotype 1 is followed by serotype 2. Okay, so what is meant by this slide is that in case a person has got infection with a serotype, okay, let's say a serotype 1, okay, then that person is immune, means to the infection by this particular serotype. Okay, means the person will develop antibodies against serotype 1. Okay, so the person will not suffer from infection by this particular serotype again. Okay, it's protected against this particular serotype. But suppose a person who has been previously infected with serotype 1, okay, he again gets infected with a different serotype, that's a serotype, let's say 2. What will happen is that it will become worse for the person. Okay, that is the pathogenesis of dengue hemorrhagic fever or dengue shock. Okay, so a person, suppose a person gets infected by one serotype, okay, he'll be protected only against that particular serotype. Okay, but if this same person gets infected by a different serotype, let's say he was infected by serotype 1 initially, and he gets infection by serotype 2 at a later date, what will happen is that it will lead to a severe dengue infection. Okay, and it is more when when a person was infected with serotype 1, he becomes infected later on with the serotype 2. Okay, so you can see that in case of the outbreak which has occurred in Delhi in 2016, okay, the persons who were infected with serotype 2, later on they got infected with serotype 4. So those persons were more at risk of, means of fatality of this disease, more at risk of having a severe dengue infection. Okay, so the pathogenic strategies of dengue is that it invades the circulatory system, causing increase in the vascular permeability. As I've told you, these viruses, they cause a lot of cytokine release. Okay, as a result of these cytokines, there will be an increase in the vascular permeability. Okay, so as a result of the increase in permeability, in the vascular permeability, the platelets will come and try to block all the bleeding points. Okay, but in the process, as a result of increase in this permeability, even the platelets also, they get there'll be means hardly any platelets left, which can which can clog these blood vessels. This can lead to disseminated intravascular coagulation, okay, which finally can lead to excessive bleeding, and potentially there will be death of the patient. This is in case of a severe dengue infection. Okay, otherwise in cases of a primary infection, the first infection, most of the people actually recover. So immune response, let's see the immune response, okay, in first infection. Okay, you please understand this. During the in case of first infection with dengue, there will be humoral and cellular immune response which can actually protect that particular patient. Okay, so humoral response, there will be an increase in the antibody neutralizing levels, serum neutralizing levels. It prevents dengue from cell membrane fusion. So in case of a first infection, there will be production of antibodies, okay, which will be protective against that particular serotype. Okay, which will be protective against that particular serotype. Okay, so these antibodies are neutralizing antibodies only against the particular serotype. But at the same time, there will be antibodies which are produced, means they are heterotypic antibodies, means they are antibodies which are non-specific. Okay, so in addition to these neutralizing antibodies, there will be antibodies which are produced which are actually non-specific antibodies. Okay, so these non-specific antibodies are non-neutralizing in nature, so they provide no protection against, in the patient's body. Okay, and cellular immune response during the first infection, it involves the dendritic cells which migrate to lymph nodes and activate T lymphocytes, and these T cells are able to fight off the first infection and enable development of memory cells, and these memory cells will enable the person to fight off any future dengue infection of the same serotype. Okay, so this is in case of a first infection. Okay, I'll tell you again, the first infection will ensure that both the humoral and cellular immune response will be in place to protect against the particular person. Okay, so in case of the humoral, there will be production of a neutralizing antibodies, but these neutralizing antibodies will be able to neutralize only the the virus of that particular serotype, okay, but not against the other serotype. So the second infection, what happens in the second infection? when a person becomes infected with a different serotype. So there's such a mechanism. The mechanism is actually not known, not very clearly known regarding dengue infection, but the most probable mechanism is as a result of this ADE, which is known as the antibody dependent enhancement. Okay, as a result of non-neutralizing antibodies. I've told you that there are antibodies which are produced during the first infection. Some of them are neutralizing, which are protective against a particular serotype, but some of them are non-neutralizing. So this antibody dependent enhancement are as a result of the non-neutralizing antibodies. Okay, and these antibodies will bind to these receptors, FCR receptors. Okay, so this will result in an immune enhancement. Okay, so in the presence of this ADE antibodies, there will be an increased rate of infection, increase in the number of infected cells, and increased production of viruses per cells. Okay, which will actually lead to severe dengue infection. Okay, so this is the mechanism. Okay, the antibody dependent enhancement. Please list down, means please this slide is very important. So when a person is infected with a different dengue virus type, okay, non-neutralizing antibodies from the primary infection, they cannot neutralize the virus. Okay, so what they do is actually they combine with the virus instead. They form a complex with the virus, and this virus antibody complex, it will attach to receptors called the Fc gamma receptors or Fc gamma R on the circulating monocytes. Okay, so the antibodies actually help the virus to infect the monocytes more efficiently. So inside the monocytes, what will happen is that the monocytes will be releasing a lot of cytokines as a result of infection by this complex. Okay, there will be an increase in the cytokine release, increase in the replication of the virus, and hence there will be a higher risk of severe dengue infection. So this is what happens. Okay, this is dengue virus. It will form a complex with the heterotypic antibody as a result of previous infection. They will bind to the FcR receptors of the monocytes. Okay, inside the monocytes, replication will occur. There will be an increase in viral load. At the same time, okay, there will be a lot of cytokines being released, which actually contributes to the pathogenesis of the severe dengue infection. Okay. So pathogenesis of dengue hemorrhagic fever or dengue shock syndrome, it's as a result of antibody dependent enhancement, okay, which can result in a cytokine tsunami resulting in movement of body fluids to extravascular space. So there's a lot of plasma leakage as well as the disseminated intravascular coagulation, increased vascular permeability, there will be hemorrhage, there will be shock. And finally, in most of the patients, death can occur. Okay. So clinical dengue, as per CDC, that is Center of Disease Control Atlanta, it can be divided into asymptomatic, it can be asymptomatic, it can be symptomatic. Okay, and in the symptomatic group, it can be an undifferentiated fever, it can be a dengue fever syndrome, or it can be a dengue hemorrhagic fever. Okay, so dengue hemorrhagic fever, when it's involving shock, it'll be as dengue shock syndrome. When there's no shock, it'll be just a dengue hemorrhagic fever. So in case of a primary infection, okay, the most it can go is here in case of a primary infection. But in case of a secondary infection with a different serotype, it can result in this manifestation, that is dengue hemorrhagic fever. You have to remember that. Okay, primary infection can lead to an undifferentiated fever, it can be asymptomatic, it can be undifferentiated fever, it can be dengue fever syndrome, whereas a secondary infection with a different serotype, it can lead to a dengue hemorrhagic fever. Okay, so what do you mean by undifferentiated fever? Here there will be only fever, it will be mild, non-specific symptoms. They do not meet case definition for dengue fever. Okay, and they are typically young children or those experiencing the first infection. They recover without need for hospital care. Okay, and a classical dengue fever, this is also relatively benign, self-limiting cause, typically in older children or adults. Okay, there will be fever 2 to 7 days, sudden onset, biphasic. Okay, headache, retrobulbar pain, conjunctival injection, okay, pain in the back and limbs, that's why it's known as breakbone fever. There'll be lymphadenopathy and maculopapular rash, okay. Even though these symptoms are there, but they may be relatively benign, okay, and the cause is usually self-limited. But in case of dengue hemorrhagic fever and dengue shock, initially there may be symptoms of dengue fever, but suddenly, okay, symptoms of hemorrhage can occur, thrombocytopenia and shock can occur. And these epidemics have been seen in many Asian countries, okay, in Thailand, Southeast Asia and India. So dengue hemorrhagic fever, how can you define this one? According to WHO, there will be thrombocytopenia, that is a platelet count will be very less because the platelet has been used up. Fever 2 to 7 days, okay. Hemorrhage manifestations with a positive tourniquet test. A positive tourniquet test is when you use that blood pressure cuff, okay. So when you use that, you leave it for some time, means you increase the pressure on the arm, okay. You use it on the arm and then you increase the pressure, you'll see tiny petechial spots, okay. If it's more than 20, that will be a positive tourniquet test. If there are more than 20 petechial spots, okay, that will be a positive tourniquet test. Or there will be petechiae in the skin, ecchymosis or mucosal bleeding. All these are hemorrhagic manifestations. And hemoconcentration or evidence of plasma leakage as seen in ascites, as evidenced by ascites or any effusion or decreased albumin. Okay, so when these points are satisfied, when these points are being means these points are there, means it will belong under classification of dengue hemorrhagic fever. Okay, so you can see these are the petechiae, ecchymosis, okay, a lot of bleeding. Okay, now coming to laboratory diagnosis of dengue, it can be hemogram, that is counting the platelets, hematocrit, leukocyte count. Virus may be isolated using the cell cultures. Serological diagnosis will help a lot, okay, because most of the labs are diagnosing using serological diagnosis, okay, and molecular diagnosis for the confirmation. Okay. Serological diagnosis is actually means suggestive. It's not definite diagnosis. Molecular diagnosis is actually the one which confirms um, dengue. Okay, it confirms. Laboratory diagnosis, there will be leukopenia, thrombocytopenia, and moderate liver enzyme elevation. So if you want to choose which test you want to do, okay, see, you have to know this. In the primary dengue infection, what will happen is that, okay, you'll see that the IgM antibody will increase, but it'll fall within 3 months. Okay, this is non-structural protein of the dengue virus. Okay, it will be found as soon as the infection takes place, but okay, it will be there for approximately 1 week or 1.5 weeks. Okay, whereas the virus will be, again, virus also will be there for approximately, if you have to do a molecular method, the virus will be there for approximately within 1 week. Okay, so if you choose antibody testing, okay, for IgM, it should be, means in case of primary infection, for IgM it should be within 3 months. Okay, you can try to detect IgM. Okay, at a later stage, if it's beyond 6, means 2 weeks, okay, 2 weeks later, you can try to test fourfold rise of IgG. Okay, but if you want to test for the non-structural one, okay, that is a component of the dengue virus, you will have to test it within 1 week. Okay, similarly, if you want to do a molecular testing to detect the virus, the collection of sample should be within 1 week. Okay, in case of secondary, all these parameters more or less the same, but you'll see the non-structural one, you can detect it within 6 days. Okay, up to 5 days. Okay, you can try to detect this non-structural one. If you use this method of detection of non-structural one, beyond this you may not be able to detect the virus. Okay, and if you do a molecular method, you can detect it within 1 week. Okay, if you're going to do a molecular method to detect the virus. Okay, IgM, again, in case of a secondary infection, you can use serological testing, okay, for IgM, but it has to be within this 3 months, maybe at 2 months. Okay, within 2 months you can try to detect the IgM. Beyond 2 months you can try to detect this for full rise of IgG. Okay, that is how you will choose the test accordingly. So you can see these are the test diagnostic process. Okay, for acute cases, okay, for acute cases you can try to identify the dengue serotypes using molecular method. Okay, whereas for the convalescent cases, okay, convalescent serum, you can try to detect IgM ELISA in case of recent infection, but in case of infection beyond 2 months, you can see for a fourfold increase in the IgG. So method, viral isolation characterization can be done, antibody detection as I've told you, and genomic sequencing. Viral isolation is actually the gold standard, but it's not possible. It's not possible because it's not rapid and it's impractical for rapid diagnosis and treatment. Cell culture of mammals and mosquitoes are used here, and the method of identifying is using indirect immunofluorescence. Okay, serological test, as I've told you, they have different serological tests. You can do, this is what is followed commonly, that is IgM capture ELISA or the MAC ELISA, which is usually done for acute cases, means cases which are within 2 months. Okay, for detection of those cases. Rapid immunochromatography test can also be done. This test is available for detection of the non-structural protein 1 of the dengue virus. Okay, antigen detection can also be done through ELISA, that is non-structural protein. Okay, can also be detected through the method of ELISA as well as rapid immunochromatographic test. Okay, so this is the ELISA method, Denmark ELISA, no, IgM capture. So you can see here that plate is lined with anti-immunoglobulin, okay, anti-human immunoglobulin. So if you add the patient serum, if it contains this IgM will bind to this anti-immunoglobulin, okay, which is coated on the sweat. Okay, then the next step, which it will be adding a dengue antigen. Okay, if dengue antigen is there, it will bind to the immunoglobulin of the patient. Okay, and finally this, you add an anti-dengue antibody conjugated with enzyme. Okay, understand, and this will be means visualized using a spectrophotometer. Okay, so this is the MAC ELISA. Molecular methods are quicker, more reliable methods of diagnosis. It can be used during acute stage of the disease. So treatment, no specific therapy, supportive measures can be there like hydration, acetaminophen if no liver dysfunction is there. And for cases of dengue hemorrhagic fever or dengue hemorrhagic fever with shock, there should be IV fluid resuscitation and hospitalization and blood or platelet transfusion when needed. Okay, so the treatment is no specific therapy, only supportive measures. That's why in the case of dengue, usually the supportive therapy is usually instituted. They do not need to know whether, because sometimes testing, we may not have the test and all, you need not wait for the test. If there's a strong suspicion of being a dengue infection, all these resuscitative methods should be instituted without waiting for the test results. Okay, so prevention can be personal, that is clothing to reduce exposed skin, insect repellent, especially in the early morning or late afternoon. Environmental means you have to reduce the vector breeding sites. Solid waste management has to be done. Public education has to be done. Okay, and biological, using these larvivorous fish like Gambusia, endotoxin producing bacteria like Bacillus. These are which targets the virus. Okay, and Copefoot crustaceans, which are mesocyclops. All these will help in taking care of the mosquitoes. Okay, and chemical, insecticide, treatment of water containers, space spraying can be done. Okay, and the vaccination, this vaccine, chimeric yellow fever live attenuated tetravalent dengue vaccine has been means used in certain countries but not in India. I think that is all. You can read the other topics